everyone. You are listening to Through Time and Clades. My name is Joan,
And I'm Albert.
and here we are with Humanity a Prologue, Season 2, Episode 2, Ancestry. It's been a long time coming here to get this episode out. But before we do that, how you been doing, buddy?
Well, it hasn't been too long since we last recorded. It's only been about a week, almost exactly a week, really. Yeah, I mean, things haven't changed too much for me in the meantime. I'm still kind of working on research projects mostly, and making, I would say, pretty good progress on those. So I'm reasonably happy, I suppose. Um, if I'm not mistaken, in this episode, you're about to introduce us to, I guess, an organism called Luca. And I was wondering if I could say some words on behalf of Luca here in this intro.
By all means, go right ahead.
Right.
My name was Luca. I lived on the ocean floor. I lived a long time before you, so you probably never seen me before. But there is something in your genes that you share with sardines and evergreens that can tell you what it means. Oh, let me tell you what it means. I think it's 'cause I'm tiny that you doubt our common ancestry. But if you look real close, you'll find me at the rootstock of your family tree. I was a life form that emerged before archaea and bacteria diverged. And from this planet I'm now purged, yet in every life form I'm submerged. Yes, even in you my seed is splurged. My name was Luca, and I am nature's stowaway. But I am so much more than just some simple strands of RNA. So if you think that you're unique, then let me shatter that mystique. But please don't think that this view is bleak. Herein lies all the truth you seek of Mother Nature's common streak. I've long since left the ocean floor, and I am here to let you know the score. Your genomes come from a collective store, so please don't argue anymore. Oh, please don't argue anymore. I am your last universal common ancestor. My name was Luca.
Hey! That's very good. Is that Professor Flint?
No, actually, that one's from John Hinton. Yeah, I think it's a parody of a song that's called "My Name Was Luca," but it's about the last universal common ancestor and not a person actually named Luca.
Oh, that's fantastic. That was very well done.
Oh, thank you.
Yes, you are correct. We're going to be talking about Luca and all of Luca's descendants, or at least tracing Luca's descendants till we get to the point where we emerge, or at least our last universal common ancestor with chimpanzees and bonobos. Um, yeah, so at long last, we are back to continue with this anthropological journey of ours. But as I mentioned in the last chapter's episode preview, we're going to shift gears and go back to the beginning of life on Earth and then trace forward our evolutionary lineage. This is to show the gradual accumulation of physical traits across billions of years that we find in the human body today, or at least until the time, again, of our last common ancestor with chimps, who are our closest living relatives. We'll pick up the story of our physical evolution from there in the next episode. And that's kind of a way to show how far scientists have come in tracing our origins and fleshing out the great bushy tree of life. I've decided to show to the left a representation drawn by
Hmm.
the German biologist Ernst Haeckel in
Mhm.
his 1879 book, The Evolution of Man. Of course, it's purposely drawn to place great prominence on humanity in particular, as was the style of the time. And it may seem like we'll be doing the same in this episode, but we must stress that there's no scientific evidence to suggest that humans are the pinnacle of evolution as depicted here. Today, we'll simply be following the path through our extended family tree to show just where exactly we are. Nothing more, nothing less. I mean, if we were doing this episode about, you know, all the traits leading up to sea stars or orchids, well, then we'd do the same thing. And so, what do you think? Are we ready to get started?
Yeah, sounds good to me.
Excellent. So let's jump to the next slide. Um, let's we'll bypass abiogenesis, the origin of life, by just mentioning that to date, we lack a major consensus for the exact processes that gave rise to biological organisms. The most recent understanding points to an early Earth and a wider universe full of inorganic and organic molecules, and that hydrothermal environments deep below the ocean's surface may have provided the energy required to synthesize these molecules into the first eventual living things. As to when this occurred, or for how long, we cannot say either. But the oldest definitive fossil evidence points to the earliest life on Earth existing by around 3.8 billion years ago, although there are older candidate fossils that are known and point to a presence of life not long after the formation of the Earth itself. And contributions from genetics further suggest this, with a recent study from just last month arguing that life has been on Earth for at least 4.2 billion years. Interesting to see where that goes in the future. Today, the traits that define all biological organisms are the presence of small self contained chemical factories called cells that use DNA, that is deoxyribonucleic acid, which contains the instructions for replication and thus the creation of new life. In fact, it is this ability to grow, reproduce, and keep our bodies maintained that is widely considered to distinguish life from non life. Now, the question of where viruses, viroids, and their kin fit into this is a story for another day. But if research from recent times is anything to go by, it may be that the last universal common ancestor of all life on Earth, the aforementioned Luca, was a far more unique population of organisms than can be assumed by simply counting all the traits that define life in the present. For example, in a summary by Madeline Weiss and colleagues in 2018, quote, genes that trace to Luca ancient biochemical pathways, and aqueous reactions to CO2 with iron and water all seem to converge on similar sets of simple exergonic chemical reactions as those that occur spontaneously at hydrothermal vents. End quote. Ancient genetics points to Luca being an anaerobic prokaryotic organism, so having much simpler cells than even modern bacteria, that metabolized carbon and nitrogen rich hydrothermal gases for energy and was thus adapted to living in a sulfur-rich thermophilic environment. Research on this front is always changing, however, with a recent 2024 paper suggesting that Luca was instead an atogenic organism, so producing acetate salts as a byproduct of anaerobic activities,
Hmm.
that may have relied on older stem organisms for nutrients. Time will tell what new hypothesis will arise from our current evidence. Now, this propensity for existing in such extreme environments is seen in some of the oldest lineages of the first two branches of the tree of life, the bacteria and the archaea. And so let's move to the next slide. These prokaryotic organisms are the oldest lineages of living things today. And for many years, the archaea in particular were thought to be just another type of extremophile bacteria, but genetic studies within the last few decades have unequivocally singled them out as a distinct form of life, with its own unique enzymatic processes, which is something seen in eukaryotic life too, those being usually seen as the third and final major group of life on Earth, containing animals, fungi, plants, algae, and all the other microbial organisms. But if we want to be accurate, it is more like eukaryotes represent a fusion of the two major
Hmm.
branches of prokaryotic life. It has been discovered that the eukaryotic cell ultimately descended from a type of archaea called Hodarchaeales, which belongs to a wider clade that has been named simply Asgard. Now, if these names sound similar to Norse mythology, that's because Norse mythology inspired them. which is, yet another story for another day. But in essence, molecular biologists have been able to discover that these archaea are distinguished by the presence of a cytoskeleton made of actin proteins, which creates a filamentous structure around the cell that provides support for the cell when alive and help reassemble it back together during reproduction. This trait was subsequently bypassed into the eukaryote common ancestor and is found in all eukaryotes today. A team of geneticists led by Laura Eam just last year have been able to show that the common ancestor of the Asgard archaea lived as thermophilic chemolithotropes, which is a fancy way of saying that they were archaea adapted to geothermal or hydrothermal environments and survived on carbon molecules. But by the time the direct archaeal ancestor of eukaryotes had evolved, these organisms had moved into mesophilic environments with more moderate temperatures
Hmm.
and had acquired genes that would be beneficial for a heterotrophic lifestyle, or that is, being able to ingest organic matter for food. So let's go to the next slide. The contribution of bacteria into the eukaryote common ancestor was via a process known as an endosymbiotic event. At some point before 2.2 billion years ago, a Hodarchiales archaea had ingested an alpha proteobacterium as food. Seemed like nothing special, but instead of digesting the bacterium, the archaeal cell kept it alive inside its body. Now, just how this happened is still being studied, but we do know that subsequent adaptations were required to allow a protein transport system to be constructed between the host cell and its bacterium. This is important because alpha proteobacteria are aerobic microbes. They require oxygen. And so the presence of an oxygen breathing bacteria within an archaeal cell can allow the latter to survive in oxygenated environments, as were emerging in greater frequency thanks to the evolution of photosynthesizing bacteria. Thus, it was that the eukaryotic cell was born, its bacterial companion evolving to the first mitochondrion.
Hmm.
Now, there are still questions being debated about when other aspects of the eukaryotic cell, like its endomembrane system or the sequestration of DNA into the nucleus, first appeared, with the most recent research suggesting that these processes may have already occurred prior to the endosymbiotic event. From there, eukaryotic life evolved and diversified, with fossils showing eukaryotic organisms in present day northern China by 1.63 billion years ago, and in Canada by 1 billion years ago. Because such fossils are rarities due to their lack of hard parts, phylogenetics have had to pull most of the weight in reconstructing relationships, with the absolute latest models pointing to yet another great split in the tree of life between a clade called Diphoda, which includes many autotrophic organisms like plants, and many groups typically called algae, and Amorphea, which included mainly heterotrophic organisms like amoebas, slime molds, a host of obscure flagellated organisms like mammaliamorphs, and a clade called opisthokonta. Now, before we continue, Albert, do you have anything you'd like to add so far?
Well, that seems like a very succinct summary of what is a huge topic, so I have to applaud you for that. Endosymbiosis is one of those concepts that's just so mind-blowing, right? Just this idea of like evolution not just proceeding in a line of solely direct genetic descent, but also kind of organisms combining to become like a brand new organism. And I guess one thing that I want to give a shout out to is the new 2024 study on Luca that you mentioned. I believe the lead author on that one was Ed Moody, who was a classmate of mine when I was studying for my master's degree at the
Oh,
University of Bristol. So yeah, it's very cool to see him get like this big, high-profile paper out. It's really amazing work.
well, that's wonderful. Well, good to hear that. So let's move on to the next slide and look at the opisthokonts. Now, these have been recovered since the earliest phylogenetic studies on eukaryotes as a natural group. And so today, Opisthokonta is robustly supported. Although it's only within the last 2 decades that genetic synapomorphies have been clarified. One thing that is important to recognize is that opisthokonts all possess a single posterior flagellum during at least one stage in their life cycle. On the top right, you can clearly see this at play. Image B is an animal sperm cell, while images A and C show a choanoflagellate. These are the closest living relatives of all animals on Earth, and they are fascinating organisms in their own right, sometimes living as free swimming creatures and sometimes forming simple colonies. And they appear remarkably close to the internal cells that sponges use in filter feeding. So maybe this is an idea of what the very earliest animal common ancestor might have looked like. And we've even learned very recently that some large choanoflagellate colonies house multiple species of bacteria within themselves,
Hmm.
which live and feed completely within an extracellular matrix. In effect, a rudimentary form of gut biome. This may have much to teach us about how the gut biomes of animals first evolved. But moving on, opisthokonts can be divided into two clades, holomycota, that is the fungus-like organisms, and holozoa, the animal-like organisms. Choanoflagellates in animals share the latter clade with a number of other obscure groups. Ichthyosporea, for example, are microscopic parasites of fish, where they form spore-bearing spheres. But unfortunately, we don't have the time to cover them fully either. So let's move to the next slide, where we thus finally arrive at the so-called animal kingdom, or metazoa. One important, while not exclusive, feature of animals is that they're multicellular for at least one stage of their life cycle. Given that choanoflagellates have propensities towards forming colonies, as indeed many other microbial eukaryotes, that this change happened is altogether unsurprising. But what is important, however, is that animals have reinforced and complicated their multicellularity through a gradual series of gene regulatory mechanisms, utilizing different cell types for different functions and evolving the extracellular matrix for cell adhesion and communication. They've also evolved a germline and oogamy, so they have cells that develop into eggs and sperm, as nearly all animals practice sexual reproduction. So it is that animals are not simple colonies of cells, but much more complex amalgamations into a greater whole. Animals have also retained their heterotrophic nature, and nearly all of them have to ingest other organisms for food. The nature of the last common ancestor of all animal life, following the choanoflagellate-like forms, has been debated with much passion. And some of you may recall that within the last decade or so, there's been a bit of a back and forth about which living animals represent the first to branch off,
Hmm.
the sponges or the comb jellies. So far, the most recent evidence favors a sponge first model, which is argued to make the most sense from a morphological point of view. Sponges are rather simple animals with cells that are not very specialized. You can even slice up a living sponge until it's a mist of cells, and they'll all reassemble back together into a whole animal.
Hmm.
And sponges lack the gastraea, or ectoderm endoderm body plan that unites all the other animals, where there is a separate outer and inner tissue layer making up the body, which allowed food to be digested within the gut, which is something that unicellular organisms have to do with their cells alone. Now, beyond this, there are still many questions about early animal evolution. For example, where did the various ediacaran organisms go on the family tree? The phylogeny that I have presented here is a rather simplified consensus of several latest studies so far, which highlight more recently evolved early animals like the Cladnidaria, these are the corals and the jellies, which have evolved proper tissues, muscles, and nervous systems for the first time, as well as contain Hox genes in their genome, which regulate how the body develops and where its constituent parts should emerge within the embryo. So let's move to the next slide. All other animals belong to a clade called bilateria, which, you guessed it, designates animals that have bilateral symmetry, at least ancestrally. For some groups like the echinoderms, that is the sea stars, urchins, crinoids, and kin, have re-evolved the radial symmetry seen in earlier animals like jellyfish. The ancestral bilaterium can be conceived as essentially a worm, and I must stress that worm is a term that describes an invertebrate animal body type that is very elongated and usually without limbs. So it's not a natural group with a scientific name,
Hmm.
as it was in the past. One can imagine an animal with the vibes of a planula, which is a flattened swimming form preserved today in the larval forms of earlier diverging comb jellies and cnidarians, but with more developed muscles for contraction and expansion of the body and movement, and retaining a single opening for ingestion and expulsion of food. Perhaps a more fitting ideal would be the living xenocolomorphs, one of which is shown here at the upper right, which have just such an anatomy. What is important about bilaterians, as opposed to the planulae of cnidarians and all other early animals, is that they had distinguished front and back ends to the body, with a head end controlled by a larger bundle of nerve cells that make up the early brain. Indeed, just last year, in a paper by Helen Schuster and Frank Hirth, regulatory gene sequences specific to the midbrain were traced to a single origin within the last common ancestor of bilaterians. Now, beyond this, things get tricky in the study of animal evolution. Traditionally, it was understood that the family tree of bilaterians could be classified with the xenocolomorph worms as the earliest diverging branch, and the one that retains the ancestral characters, followed by another split where there's an acquisition of a complete gut that threads through the entire animal. The lineages would have been divided into those where the mouth opens in the embryo first, protostomes, and those where the anus opens first, the deuterostomes. Now, the protostomes would include the vast majority of invertebrate animals, arthropods, mollusks, flatworms, nematodes, brachiopods, bryozoans, and more, while the deuterostomes would include the echinoderms, the acorn worms, and graptolites, like the one pictured at the bottom right, as well as chordate invertebrate animals. Recent studies on animal phylogeny have begun to cast some doubt on this neat picture, however, for it may be that the deuterostome condition emerged first in bilaterian evolution, and that the protostome condition was a derived feature in one clade of subsequent animals. Add to that the inconsistencies between many of these studies that find xenocolomorphs as either to be early bilaterians or close relatives of echinoderms and acorn worms, which are in their own clade known as ambulacraria. More work needs to be done before this can be properly confirmed. So for now, I show the bilateria subclade here as a polytomy. But there's one more small controversy that needs addressing. Now, some of you may be familiar with the early Cambrian animal Saccharydus coronarius, which was described in 2017 by Jean Hahn and colleagues, and promoted by Simon Conway Morris as a particularly special animal that was the oldest known deuterostome and thus a representative for what the common ancestor may have looked like. I have it illustrated to the bottom right here. Well, subsequent work has shown that this animal was in fact an early type of protostome, specifically allied to the ancestors of arthropods, nematodes, and their kin. So maybe it's special in a different way, but not necessarily with our immediate ancestors. Is there anything else that you'd like to add, buddy?
I mean, not too much in particular, but these phylogenetic problems regarding kind of the deep divergences within animals is definitely a topic that I'm interested in. And in fact, one I have an indirect connection to because when I was studying for my master's degree at the University of Bristol, my supervisor, Professor Davide Pisani, he's someone who has done a lot of work on like phylogenomics, so using genomics to study the phylogeny of different organisms, including animals in general. And he has published a number of things on the sponges versus comb jelly or ctenophore debate. His publications mostly come down on the side of being in favor of sponges first, as you have portrayed here. But I know that there have been some recent studies that have still continue to promote the ctenophore first model as well, which I know other people are prepared to rebut. The deuterostome debate I feel like it's a bit more recent, or at least one that's been called into question a bit more recently. But I'm definitely very curious to see where that goes too, because for a long time it seemed that the deuterostome versus protostome split was very well established, and yet now we have a few studies questioning that. So, yes, definitely a lot more work needs to be done to tease these problems out.
Oh, certainly. It makes you think about, what well-established results in phylogeny may be called into serious question in the coming years.
Hmm.
Things that we're pretty used to seeing as well supported. So who knows? Who knows? It's very fascinating indeed. So let's go to the next slide now. Thanks to two recent papers by Giovanni Mussini and colleagues and Thurston Licali, we know we now have much more up-to-date information about this part of our family tree. Perhaps unsurprisingly, the vertebrate-invertebrate dichotomy in animal classification is more of a convenience for, of course, invertebrates involved from invertebrate animals. When we look at the clade chordata, we see a number of fossil living forms that showcase how the vertebrate body plan formed. The last common ancestor of chordates possessed a notochord, which is a flexible shaft that runs along the dorsal part of the animal's body, which allows a hollow nerve cord to run through. The back end is also more distinguished, having a segmented posterior section, in other words, a postanal tail. Among living chordates, the notochord is retained only in the earliest diverging groups, the fish-like lancelets, and in larval tunicates, which include things like salps and sea squirts, as well as in the hagfish, which is a special case. Vertebrate animals lose the notochord in embryonic development when it calcifies and becomes the vertebral column, which makes it a later evolutionary change. Chordates also ancestrally have pharyngeal slits for housing gills to better breathe oxygen in the water. Likewise, for the notochord, this is a feature that has been lost in subsequent lineages, primarily those that took to a land living existence. But even in these, they're still present in the embryonic stage. In the fossil record, we find now extinct clades that further bridge these gaps and show, when each of these different features emerge. The first to branch off were several mysterious clades that have been previously grouped together as vitulacolians. These were bizarre fish-like animals that only lived throughout the earlier half of the Cambrian period. They had a large front half and a smaller tail, like some aberrant tadpole. And I have one pictured here at the top right. It's only with this recent paper that their phylogenetic position has been better clarified, since they've been interpreted in the past as belonging to other groups, even early relatives of arthropods in previous studies. After these groups branch off, early chordates extended the segmentation of their tails to the rest of the body, and this change is seen in the next clade to branch off, the Cambrian Unanozoan. Then chordates evolved inflected myomeres, which are segments of muscle tissue supported by skeletal, or in this case, protoskeletal elements. So if you've ever bisected a fish or cooking, then you've seen this arrangement. The next group to branch off, and the earliest to show these traits, is another Cambrian organism called Pikaia, which is illustrated in the middle right in black and white. The reinterpretation of Pikaia fossils by Mussini et al. As definitively showing a dorsal nerve cord and gut actually showed that prior restorations had flipped the animal upside down, which is a phenomenon that is not unheard of in Cambrian animals. If I had a nickel every time a Cambrian animal was portrayed upside down in his initial publication, I'd have 2 nickels, which isn't a lot, but it's weird it's happened twice. Of course, I am, of course, referring to the early relative of velvet worms, hallucigenia, which is another fun story to talk about. Next, chordates fully developed their postanal tail, and it's thus that the first of the living clades branched off to become the lancelets, represented today by about 30 tiny species that burrow in seafloor substrate and filter feed in the water. Then chordates evolved a more refined olfactory and respiratory system in their pharyngeal cavity, which allowed for greater senses of smell and more efficient breathing. We also see the development of the neural crest, which are transient cells in embryonic development that then grow into a wide variety of tissue types, which ultimately paved the way for the evolution of cartilage, bone, skin pigmentation, smooth muscles, and a more elaborate nervous system. The inheritors of these traits gave rise to the tunicates on one side and the craniates on the other, who then evolved calcified cartilage around the organs of their head end, and so gave us the first skulls. We also find, based on embryonic studies, that true camera eyes first entered the chordate body plan. In earlier organisms, the possibility for eyes started with simple cells that could distinguish between light and dark, followed by adaptations that dipped the light detecting cells inside the body So now organisms could distinguish distance as well. And with the later development of rhabdometric photoreceptors, now organisms could actually see their surroundings and process it in their brains. As seen in hagfishes and larval lampreys, these being the oldest vertebrate lineages around today, the ancestral craniate eye would have been such an eye, and having a proper retina and ganglion cells for enhanced vision processing, and an optic nerve connecting it all to the brain. There even would have been a translucent bit of skin in the front of the eye to protect it, a sort of proto-lens. Craniates ultimately gave rise to vertebrates, and their nearest sister relations have been found in the Cambrian fossil record, these being the three species of mylocynmingids, which could fairly be called the oldest known fishes. I have one shown here at the bottom right. For you fans of Walking with Monsters, the animal Hycoichthys belong to this group. Mylocynmingids have been interpreted as free-swimming animals, with early fins along the posterior end for propulsion, just like fishes use today. So let's move to the next slide and talk about vertebrates. Now, fish evolution continued with the development of the vertebral column from the notochord through calcification of the rod-like organ into a cartilage backbone. This is reflected today in the oldest living lineage of vertebrates, the cyclostomes, or the hagfishes and lampreys. Although the hagfishes seem to have reduced their vertebrae into small nodules while still retaining the skull. They have been shown to be related to two fossil groups, the similarly eel-like chordates and the anapsids, which have diminutive mineralized scales. And this is an echo of things to come, for the basalmost family tree of vertebrates is full of a number of now extinct forms of jawless fishes that were common elements of the marine fauna through the Ordovician and up until the Devonian mass extinctions. Some examples of these, of the pteraspidomorph clade, are shown at the right here. The majority of these early fishes had ossified dermal armor covering most of their vital organs, at most allowing their eyes, noses, mouths, and fins access to the outside ocean. While some were among the first vertebrates to colonize pelagic waters, most seem to have been poor swimmers that kind of hovered along the seafloor and sucked up food with a simple mouth. Paired pectoral fins, common to most living fishes today, seem to have emerged within the common ancestor of the osteostracans and the gnathostomes, or the jawed fishes, as anticipated by older fossils of the related galeaspids, who were recently discovered to have had ventrolateral fins that ran from head to tail. So it was only later that this separated into the anterior pectorals and then later posterior pelvic fins, as seen in jawed fishes and their relatives. This change was also followed by a rearrangement of the head and mouth organs. It was previously hypothesized that the transition from a jawless plated sucker mouth to a mouth with hard parts or denticles, that is, proto-teeth, was facilitated by adaptations towards more active predation. But new studies on pteraspidomorphs shows that jawless fishes evolved denticles in their filter-feeding mouths long before this change. It seems that instead teeth initially evolved for keeping materials from getting lodged between their plated mouth organs, and only later were repurposed for biting and tearing prey animals. Let's move to the next slide. The origin of jaws remains to this day an area of debate among paleontologists and biologists. The traditional hypothetical model since the 1870s was that the forwardmost gill arches, now having moved internally from the gills, gradually formed the maxilla, mandible, and supporting hyoid arch, meaning that jaws essentially evolved from gills. This model had been contested in recent years because there is no evidence from the fossil record showing such a change. Contributions from comparative anatomy, developmental biology, and new discoveries in paleontology have somewhat clarified the picture, showing that there is a shared developmental origin of gill arches and jaws. The most recent hypothesis is that the velar cartilage of jawless fishes, so that's a hinge-like structure used to aid in both feeding and breathing at the back of the mouth, developed into the mandibular arch through the changing of neural crest cells. This mandibular arch was of a large and substantial size compared to the other gill arches, and seems to have undergone a division that created a new gill arch to sit between it and the hyoid arch, this being the spiracular region, which was involved with the evolution of spiracles for aid in breathing. And this region developed a small organ called a pseudobranch, which was removed from its role in breathing and now supported blood flow to the eyes. Many of these changes would eventually be lost in subsequent lineages, but the rearrangement of these facial cartilages paved the way for the maxilla and mandible to form. The ancestral gnathostomes now had movable jaws with which to secure food and a more complex respiratory system. Conventionally, the earliest jawed fishes were all considered members of a single group called placoderms, which inherited the plated body armor of their jawless predecessors and spawned some truly impressive forms, things like Bophiolippus and Dunkleosteus. But recent studies show that placoderms are best understood as an evolutionary grade of successive clades that branched off before the common ancestor of the living jawed fishes. We now know from recent fossils that some of the earliest placoderms, like the bottom dwelling antiarchs, possessed the first proper pelvic fins as discussed beforehand. By the time that genera like Entelognathus emerged, and I'm showing that here at the top right, jawed fishes had evolved anal fin spines, which are retained in early relatives of sharks, but later lost in subsequent forms, as well as in bony fishes, as well as proper scales, which would eventually become the main body covering after the body armor was lost. And although it's unclear just when this happened in early gnathosome evolution, we can be confident that by this point, the modern three-layered retina eye with a cornea and iris had evolved, allowing for the modern camera eye that we have today with all its pros and flaws. And then there's a major split between two groups. One is chondrichthyes, which are fishes that retained cartilaginous skeletons and include the sharks, rays, deep ocean chimeras, and a host of now extinct spiny fin forms known as acanthodians. And the other is osteichthyes, which are fishes that, at least initially, retained cartilaginous skeletons as well, and these were kept in the living bichirs, sturgeons, and gars, but at least twice within the group ossified their skeletons into the first true bone. Early bony fishes like Saurolepis and Wayu, shown at the bottom right, were still relatively unspecialized and similar to the earlier jawed fishes, but eventually we find another major evolutionary split among them. One line developed bony spines to support their fins, becoming the ray-finned fishes and spawning the overwhelming majority of living fish species today. Of course, it's of these that the teleosts would develop true bony skeletons for the first time, while the other developed bones within their fins supported by strong muscles, becoming the lobe-finned fishes, and our ancestors are found among them. Is there anything that you'd like to add?
Well, pretty much all of what you've just described is definitely another area where we have seen many major scientific advances in recent years, as you've kind of shown in your presentation, because you referenced some very recent studies. Yeah, I remember when I was in college, but I think like the actual like first studies to demonstrate this were coming out like before I was a college student even. But yeah, like I remember living through a time when um, we started thinking that placoderms were not a real group and that they were, as you show here, a grade kind of leading up to the modern jawed vertebrates, and that was a pretty big shift, like in our scientific understanding in my lifetime. Um, and yeah, you mentioned earlier the recent study on Picaya, and you can see like how many new advances are really changing our understanding here. I noticed that the black and white illustration of Picaya that you pictured, I think that is based on the old restoration where it was upside down, right? Because I think the new one actually has it like flipped over from what that one shows.
Oh, oops.
One of the tricky things about like this part of the tree is, of course, the fact that we don't have like a bony internal skeleton until within the modern bony fishes. Um, and earlier on, you have things like the placoderm grade jawed vertebrates and things like osteostracans that do have bone, but since their bone only covers the outside of their body and like basically only part of their bodies at that, it can be very difficult for us to get insight onto what the rest of their morphology was like. And it's really only with very exceptionally preserved fossils that also have the soft soft tissue that we understand the evolution of things like the origin of paired fins and such. And of course, that goes double for things like stem chordates that don't have any bony elements at all. And so it is quite remarkable how many recent advances we've gained in this part of vertebrate evolution in recent years. And I'm sure we'll be privy to many more.
Oh yeah, it's really impressive to me because I'm so used to like seeing studies that change the external appearance of like dinosaurs and
Mhm.
pterosaurs. But to see that the same research is going on with jawless and jawed fishes of like
Right.
the Silurian and Devonian, um, it's neat to see because you'd think like we'd have more complete fossils of these animals that we'd pretty much have the restorations down to a T. But there are still surprises even here, and it's really encouraging to see. So let's then move to the next slide. Um, and it's here where, again, I'm stressing many things, but it should be stressed before going forward that our lungs were not specifically evolved for land based living. Early bony fishes had lungs, and even if they used gills for breathing in water, the lungs were also still used for breathing. This, technically, you'll see this in the literature referred to as an air filled organ, was used in early bony fishes for buccal pumping, where the animal opened its mouth at the water's surface for additional oxygen from the air, especially in areas that are oxygen poor. Many modern fishes still do this, that still have lungs, so like bichirs, gars, and lungfishes,
Mhm.
and it's only later that teleost fishes turned this into the swim bladder for controlled buoyancy, while the lobe-finned fishes retained the lungs and just refined them. But again, that organ predates terrestriality and is found in the common ancestor of the bony fishes. Okay, moving on. The lobe-finned fishes encompass a vast array of forms that were for a time one of the dominant predators of the world's waterways, but are today only represented, in your archetypical fishy form, by the deep ocean coelacanths and the freshwater lungfishes. Tetrapods, that's amphibians, reptiles, birds, and mammals, are also members of this group, with a lot more legroom, no pun intended. And it seems odd to think about, fishes crawling out of the water and onto land being the ancestors of terrestrial vertebrates. Don't fret. Our fossil record for this gradual transition is at a very good place at the moment, with a number of key genera showing when certain features emerged. The common ancestor of coelacanths, and that includes extinct forms typically called onchiodonts, and rispidians, which is everybody else, had evolved a hinged intracranial joint, which gives the jaw a more enhanced bite force, as well as a single bone that was attached to the shoulder and hip joints, becoming the humerus and femur, respectively. This gave the limbs more flexibility of movement, which, aided by strong muscles, allowed lobe-finned fishes to colonize more regions, especially crowded freshwater rivers and streams. Then we see a split between the lungfishes and tetrapodomorphs, which evolved an autostylic jaw suspension, which is a fancy way of saying that the upper jaw or maxilla became attached to the skull. Then, with the splitting off of the early Devonian canictes, we find the origin of the choana, or internal nostril, among the tetrapodomorphs. This was a major development in the respiratory system, because now there was a connection between the nasal cavity and the throat, which is a more efficient way of bringing air to the lungs, because now the animal doesn't have to gulp or swallow the air in through its mouth. Next, there are a number of splits by now extinct lineages, including the giant predatory rhizodonts and the smaller osteolipidids, in which a distinct socket developed for the head of the humerus to fit into the shoulder girdle. And now we approach a notable series of so-called transitional forms that showcase ever more new traits that will go on to define the living tetrapods of the land. First, there are forms like Platycephalichthys and Eusthenopteron, the latter is down at the bottom, at the top right, which gained further changes to their humerus as well as at the back of the skull. While popular paleomedia over the decades has depicted Eusthenopteron in particular as, an important lobe-finned fish that frequently made trips onto the seashore, today they're more accurately known as primarily aquatic animals, the most ancient of the Eotetrapodiformes still to live mainly in this way. Following these are Elpistosegalians, like Panderichthys, in which the dorsal and anal fins are lost, and the pectoral and pelvic fins are now of a more modern arrangement of the upper limb bones. And of course, there's the now popular Tiktaalik, and I've shown that at the top right, which takes these adaptations even further, showing weight-bearing elbows, bending wrists, and the first honest-to-goodness neck. In animals like Tiktaalik, it has become clear that while still aquatic in many ways, it was becoming more and more reliant on environments beyond the water surface, apparently having spiracles like its ancestors for breathing air, and needing adaptations to better access that resource while still keeping wet. It's also apparent that opisthokonts were no longer relying on their tails as the main source of propulsion. Now that job was given to the limbs. Then we come to the common ancestor of the stegocephalians, which are starting to look less like traditional lobe-finned fishes and more like weird amphibians, which wouldn't be far off the mark. By now, the radius and ulna were at equal lengths, and distinct bone digits had begun to emerge on the limbs, as seen in forms like Elginirpaton and Ventostega. In the later Acanthostega, the forelimbs and the hind limbs were now fully attached to the rest of the skeleton, and the tough scales that had covered our fish ancestors for millions of years had given way to fleshy, porous skin. These animals also had interlocking vertebral pegs that helped stiffen the spine and gave their bodies more leeway for being carried off the ground by the limbs. Now these were far more efficient at moving on land, at least for short periods. In Ichthyostega, shown here at the bottom right, the gills have now become fully lost, and whatever there was of a tail fin was no more, and the lower arm bones had gained an olecranon process for muscle support on weight-bearing arms. And by the time of the common ancestor of tetrapods and buthetids, who are a carboniferous predatory group, the digits on the hand had become standardized to 5, which was the blueprint for subsequent adaptations of land tetrapods to come. Let's move to the next slide then. And where's Darren Nate? She feel right at home right about now. Yeah, so tetrapods, as typically defined, are four-limbed vertebrates. But obviously, this is an imperfect definition because, one, many living tetrapods have secondarily lost their limbs, and two, earlier lobe-finned fishes were also four-limbed vertebrates, but are not members of this clade. So nowadays, researchers tend to stick with a crown-based definition. A tetrapod is a descendant of the last common ancestor between living amphibians, or lissamphibians, and living amniotes, that is, reptiles and synapsids. That said, the ancestor of crowned tetrapods had unique traits of its own to distinguish it from earlier forms, like the reshaping of the first two neck vertebrae into the atlas and axis bones, which further increased the range and flexibility of movement for the neck. And it's here where things can get a little bit tricky. It has been commonplace for decades to describe early tetrapods as amphibians. But in a world where clistic terminology is becoming more and more preferred, biologists have had to redefine what we mean by amphibian. So today, true amphibians are represented by the Lissamphibia, frogs, salamanders, and the legless caecilians. So these are seen as the true amphibians as opposed to many other now extinct groups. A commonly supported hypothesis, with some fossil support, is that modern amphibians belong to a larger clade known as the temnospondyls, which flourished in the Carboniferous and Permian periods as major semi-aquatic and land predators, sometimes growing over 5 meters long. In contrast, we belong to the clade Reptiliomorpha, in which the premaxilla, or the very front of the upper jaw, had become reduced in size, which in turn shortened the length of the skull. There have been problems in classifying the early members of the group, with the arrangement shown here being a recent proposal. These extinct animals superficially resembled both amphibians and reptiles in many ways, and that's kind of obvious. Yet another brilliant example of how evolutionary transitions are supposed to look. In fact, in the past, all these forms had been collected into grades of evolution called either anthracosaurs or cotylosaurs, with emphasis on the more saurian nature. In either case, as lineages diverged over time, these animals eventually became more primarily terrestrial than aquatic, but they still seem to have needed to return to the water to at least reproduce and lay eggs. Eventually, we reached the common ancestor of the amniotes and extinct diadectomorphs, which supported two reinforced vertebrae within the sacrum, the point in the hip, where the pelvis connects to the spine, And this gave so much more support to the hind limbs that the possibility arose for larger and heavier animals to evolve. Let's move to the next slide now. The last link in the break with aquatic habits as a key livelihood was the development of the amniotic egg. Previously, eggs were soft, jelly-like structures that needed support from water, not just developed to be fertilized, as our aquatic ancestors practiced external reproduction, in which the sperm was sprayed onto the eggs in water. But with the evolution of the amniotic egg, the situation was different. The eggs had to be fertilized internally, and so the penetrative type of sex that we use was ultimately born. The egg remained jelly-like inside the mother's body, but when the time came for it to be laid, the egg gained a tough outer shell that was still porous to allow air to flow. Inside this shelled egg, the embryo was nurtured with its own water supply, amniotic fluid, inside a sac called the amnion, along with the allantois for removing waste, and the chorion, which covers all of this. And that gave the developed young all the nutrients and protection needed in the new terrestrial landscapes that early amniotes were inhabiting. In fact, with a hard-shelled egg that carried its own water, young could be born anywhere, whether in wet or dry habitats. And for the newly hatched young to survive here, subsequent changes occurred in the body as well, where the first amniotes had a thicker skin that was keratinized, made of tough, fibrous proteins called keratin, which kept water inside and the body dry outside. Now, the evolution of keratin paved the way for the future possibility of scales, hair, feathers, claws, and hooves. In fact, early amniotes would have already had claws, which has been suggested to be linked to nest digging behaviors. No longer using gills and relying more on the lungs for breathing, amniotes also developed a rib cage that could expand and contract with each breath. This is known as costal respiration. And a recent 2022 study even argued that the amniote common ancestor was endothermic, or warm-blooded. But subsequent papers challenged this proposal, which was facilitated with missing data, unfortunately. And so it's now reestablished that these animals were indeed ectothermic, or at least cold-blooded initially. As indicated on the phylogeny to the left, amniotes diverged into two main groups: the therapsids, or reptiles proper, which includes dinosaurs, which includes birds, and the synapsids, which include the ancestors of mammals like us. Just like with amphibians, the term reptile has been localized and properly defined in recent years and is now restricted to our sister group, the Sauropsida. So we did not evolve from reptiles, as often been told in popular paleo media, though the amniote common ancestor, as shown by these two early sauropsid and synapsid species to the left, would have looked superficially lizard-like. You can properly say that lizards today retain a very old ancestral amniote body plan, but they are far from primitive animals. And as well, our synapsid ancestors are no longer referred to as mammal-like reptiles either. The term proto-mammal for these stem lineages leading to the living mammals can be used in its place as a grade term. Now, that said, it's also a misnomer to say that we have reptilian brains within our mammalian brains, meaning that behavioral complexity in humans emerged through a layering of new parts of the brain over old ones. This is an old idea that often gets touted by psychologists in recent times, but this is based on an outdated understanding of neural anatomy. As Joseph Cesario and colleagues explained in a 2020 paper that I highly recommend, the cerebral cortex, that is the outermost region of the brain that has been associated with cognition and perception, is not confined to mammals, but is found among all vertebrates, and perhaps even their nearest relatives. Not to mention, it has become very clear that complex cognition is something that has evolved independently across the tree of animal life, and matters little on adding so-called new brain layers to make it more complex. The brain is a mighty organ that is still being studied extensively, but biologists no longer consider its specific anatomies to be indicative of or responsible for things like rationality versus irrationality, impulse versus discipline, and so on. Is there anything that you'd like to add, buddy?
Not much. This was a very good overview of the kind of tetrapod colonization of land. When we're looking at evolution, of course, a lot of the time we're looking at quite gradual changes over time, and I think it's quite difficult to objectively say, okay, like this was a major innovation that happened right at this point, but like something like the amniotic egg, even though we don't know too much about its origins because that's the major components aren't easily preserved, um, that must have been like this kind of major evolutionary innovation that allowed for kind of the complete divorce of the reproductive cycle from bodies of water, and that's yeah, it's quite a remarkable process to think about. I also think it definitely can't be understated, like just how big some of the early tetrapod relatives were. Like you mentioned how a lot of extinct amphibians and stem tetrapods were basically apex predators in their environments, and that's true because when you look at modern amphibians today, of course, all of them are very small. Even the really big ones, like they're like relatively small animals. Um, but things like Tiktaalik or some of the extinct temnospondyls were they could get massive. Like Tiktaalik was I think we have specimens that are up to almost 3 meters long, 10 feet, something like that. So these things would have been basically less like salamanders in terms of their ecology and more like crocodiles almost. Of course, they did have many land living vertebrates to prey on land, so probably they were preying on large arthropods and things like that. Definitely a very interesting period of vertebrate evolution, just like the rest of it is.
Oh yeah, I think a lot about the Triassic period, like when the first dinosaurs evolved, and they were sharing a world where the waterways were populated not just by like giant crocodilian-like reptiles, but also like giant amphibians like Mastodonsaurus, which it would be fascinating to consider like what evidence we have for like what the ecology was doing at the time. Like, were these giant aquatic animals like competing with each other? Did they have like niche partitioning? Were they able to kind of like do their own thing and not really bother each other? Um, well, I'll have to think about that. I'd have to do some more reading on. But yeah, very good points all around. Um, so let's move to the next slide then, and we'll enter the synapsid world. Members of these synapsida have typically been associated with a change in hind skull anatomy, the acquisition of a single temporal fenestra. This is an opening behind the eye socket, or orbit, between the squamosal and postorbital cranial sutures, in which jaw muscles attach to aid in biting and chewing. This is in contrast to the reptiles, which may have no temporal fenestra or have two of them. Moving further down the skull to the jaws, we also see changes in dentition. In their ancestors and inherited by the reptiles, the teeth may be of different sizes but otherwise retain the same shape. They are homodont teeth. Early synapsids had a similar arrangement, but it was clear that their teeth were beginning to change into a more heterodont condition, changing their shapes to suit different purposes. And this is ultimately how we got incisors, canines, premolars, and molars in our mouths. Several strange and bizarre animals evolved among the early lineages of synapsids. The famous sailback Dimetrodon Nedathosaurus, often grouped with Mesozoic dinosaurs in popular culture despite being early Permian synapsids, belonged to these groups. So you are more closely related to a Dimetrodon than a Dimetrodon is related to a Spinosaurus. But many others looked more conventionally lizard-shaped, like this Ophiachodon, shown at the bottom right. In traditional parlance, the synapsids that branched off before the therapsid clade were termed pelycosaurs. But as with many of the other names we've mentioned in this episode, that is no longer used scientifically. Let's move to the next slide now. It's with the therapsid common ancestor that our ancestors began to become more recognizably mammalian. For one thing, the limbs became more oriented underneath the body, so instead of dragging their bellies along the ground, therapsids could raise them and support themselves fully on their limbs. This would have ramifications for their respiration too. Just like the ancestors of dinosaurs, therapsids escaped from the burdens of side-to-side locomotion, which caused their lungs to breathe, one at a time, and could now use both lungs simultaneously to take in air while moving in an up and down motion, and that made them more efficient walkers and runners over long distances. The temporal fenestrae also became closed during this point, being covered by the sphenoid bone, which now took over the main duties in anchoring jaw muscles. While therapsid phylogeny is still a bit of a mess right now, we can nonetheless see a gradual change in morphology into more active, energetic animals, befitting what we'd expect to see in the ancestors of mammals. The carnivorous saber-toothed gorgonopsians were therapsids, as well as the dicynodonts, who were stout-bodied herbivores with beaks and often tusks. It is from the ranks of the Permo-Triassic therocephalians that we find a clade cynodontia of interest to us next, here in the next slide. So, cynodonts, or the dog-toothed animals, emerged during the late Permian period and survived the Great Dying into the Mesozoic era, where they eventually gave rise to the ancestors of mammals. So it is correct to say then that mammals are cynodonts. This common ancestor had by now developed a near mammalian dental arrangement and supplemented this with a bony secondary palate that separated the nasal and oral cavities. You can feel this as the hard palate at the roof of your mouth, which would become essentially and especially important in a later adaptation. As well, cynodonts evolved double occipital condyles. These are projections at the base of the skull that aid in movement with the neck vertebrae. In fact, it is with cynodonts that the mammalian configuration of 7 neck vertebrae was established, as well as the distinction between the thoracic and lumbar vertebral columns, being the chest and lower back vertebrae, respectively. And this change suggests that the diaphragm, or the muscle that assists in getting air in and out of the lungs, had evolved by this time, as it separates the thoracic and lumbar regions of the body. But the biggest changes were to occur with hearing. The post-dentary jaw bones, so those furthest to the back and away from the teeth, were reducing in size and eventually became the bones of the middle ear, the malleus, incus, and stapes, which transport vibrations in the air to the fluid inner ear, where they then can be interpreted more efficiently into sound. This would be supported by the creation of a new joint that would gradually separate these ear bones from the mandible, or eventually within the family tree of the true mammals, the jaw would be a single bone, with the middle ear bones completely detached and surrounded by the auditory bulla within the temporal bones of the skull. Is there anything that you would like to add here?
Hmm. Well, definitely another very interesting chapter in our evolution. I think in studying like non-mammalian synapsids, you really get the sense of how unusual mammal anatomy and mammal biology is, because so many things that we think are normal, are not. Like they are mammal things. Things like having a diaphragm or um, the having the three middle ear bones, or having just a single bone like making up uh, the lower jaw. All of these things are not widely present in animals in general. They are very specific to our lineage, and it is very cool that the fossil record has given us so much insight into the origins of all of these structures. Once again, we're seeing some maybe I think to some people counterintuitive patterns in the evolution of mammals, or at least the origins of mammals, because I feel like a classic idea is that oh well, our ancestors were small things up until the non-bird dinosaurs died out, and it was only after that that our lineage kind of became more diverse in terms of our size and ecology. But if you look to before the dinosaurs were even around, actually synapsids were some like the main apex predators and even large herbivores in a lot of their environments. And eventually the larger synapsids died out, and leaving only the small mammals and mammal ancestors that and they would stay like that for a while. But you can see the kind of rise and fall of these different lineages, at least when you look from the perspective of body sizes. Is not a given. It's not a constant. The history of life is a very rich one. It's quite nice to be able to appreciate it in this way.
Absolutely. And it's kind of neat to see, you know, up until this episode, through time and clades, we've been, you know, talking about many of these organisms that we've since bypassed on the path leading to human beings. And it just kind of goes to show, that not only is all life related, but life is incredibly diverse. And the path that we're following is just but one route that living organisms have taken on the Earth. And, again, if we were to do an episode like this on, say, orchid evolution, or like the evolution of, going into the reptile tree, going all the way to the birds, which Albert, of course, covered very beautifully in Dinosaurs, the second chapter, we'd see all new changes and all new developments. And we wouldn't really necessarily be even thinking about mammals, for example. We'd be focusing on that lineage and why those traits are important to those members. And so, again, like this is emphasizing that, there is no like, pinnacle or goal of evolution that leads to the human being. 'Cause each organism follows its own journey just like this. And it really kind of brings the point home that like we are all related. And that's something that I think more people should recognize and understand. So let's move now to the next slide. And it's at this point that we should really stop to consider those quintessential mammalian traits of endothermy and fur that do not have very clear evidence in the fossil record for their time and place of emergence. While it may be that endothermy was a gradual development from the dawn of the therapsids, recent work by Ricardo Araujo and colleagues from 2022 suggests that adaptation evolved abruptly during the latest most branches of the mammaliamorph clade, and so not with the common ancestor of Mammaliamorpha. This was based on a comprehensive comparative analysis of preserved ear bones, so the shape of which is correlated with the efficiency of motor coordination and spatial awareness in endothermic animals. And fur is a little bit trickier. So the oldest fossil evidence for fur actually comes from coprolites of a late Permian carnivore of some type, suggesting that at that point in the therapsid family tree, this feature emerged. Um, but just when and with what common ancestor we cannot be sure. Studies of therapsid skulls, including those of early cynodonts, do not really show evidence of whiskers, and whiskers do leave telltale marks of their presence on bone. But at least by the time of the common ancestor of the tritylodontids and mammaliamorphs, whiskers, and thus perhaps fur, would be present. And as a feature that has an association with whiskers, it's probably at this time that our flexible nose evolved. In this case, a rhinarium, or the wet nose, seen in most mammals. One trait that has a very definitive link to the mammaliamorph common ancestor are the epipubic bones, which have a number of uses for assisting locomotion and supporting the torso, as well as multicusped cheek teeth, which provide more surface area for chewing food. As these features suggest, these animals would have no longer resembled earlier synapsids with their kind of pseudo-reptilian demeanors. These were now very mammalian-like organisms, and they share their origin with that of the dinosaurs during the Middle Triassic period. This is why you'll see some paleontologists say that the story of the dinosaurs is also our story.
Mm.
Representatives of this time are represented by fossils such as Adelobacillus and Morchanucodon, shown in the top right, and they represent the end process of a gradual shrinking of body size that began with later cynodonts and ended with forms that could perch on your thumb. So let's move to the next slide now. True mammals are considered to belong to the crown group, which encompasses the most recent common ancestor of living monotremes and therians, or the live-bearing mammals. There are four more key traits to mammals that need to be considered here: mammary glands, teeth that are replaced only once, pinnae, or ear flaps, and kneecaps. The same genes that are linked to body hair are also associated with milk production, and this has led researchers to propose that the common ancestor of mammaliamorphs and tritylodontids between 246 and 240 million years ago had already evolved those traits. As to how mammary glands and milk production evolved, there are several hypotheses, mainly tied to the observation that apocrine glands, which are mainly responsible for sweat, include mammary glands. Now, in living monotremes, like the platypus and the echidnas, milk is secreted not from nipples, but from special pores in the skin that are lapped up by the young. And ancestral crowned mammals may have nursed in the same way. One hypothesis suggests that milk emerged from sweat glands as a way to supplement egg yolk as a main food source for newborns, and there's genetic evidence that vitellogenin, or genes for producing egg yolk, would have been lost after this trait had evolved. The increased use of milk consumption may have firmly established diffidonty, in which mammals have only two sets of teeth, with one generation being lost at weaning, which is replaced by a permanent second generation. Fossil evidence shows that early cynodonts had the ancestral condition of teeth constantly being replaced, and gradually across mammaliamorph evolution, more and more types of teeth were being lost and replaced only once, so that by the time of the eutherian mammals, the modern tooth condition was established. Ear flaps became a genetic possibility once the middle ear bones were fully separated from the jaw, and so it's likely that by the time of the common ancestor of the crown mammals, pinnae had already evolved. The kneecap or patella helps with locomotion and gives leverage to the hind limbs to allow for more extension and exertion. Comparative anatomy shows that crown mammals first evolved kneecaps, with birds developing them independently. The three living groups of mammals, monotremes, marsupials, and placentals, are all but a small fraction of the total diversity of the group that is now unfortunately lost. In fact, one lineage, Allotheria, which includes the multituberculates, was alone in surviving the Cretaceous max extinction event, only to die out during the Eocene epoch. Even among surviving clades, there were losses. So monotremes belong to a larger group called the Eunotheria, which includes a number of genera, including Schunotherium and Oscibotrinosspinos, which were widespread globally, instead of the platypuses and echidnas that are now confined to Oceania. Marsupials, like the kangaroos, wombats, and opossums, belong to a larger group called Metatheria, which was also global in distribution and may even have been North American in origin. The sparacidonts, like the saber-toothed Thylacosmilus, which we've covered on the show before, would not have been true marsupials and only close relatives. So if we move to the next slide, we will see, in contrast, the placental mammals are but one group among a larger clade called Eutheria. Now, confusingly, the placenta is not a defining trait of placental mammals. Marsupials indeed have one too, and they also technically give live birth. What makes these ancestors of ours so special is that their placenta has a more complex anatomy, and it nourishes the young for a much longer period of time. This is helped by the fact that the epipubic bones became lost in placental mammals, which causes marsupials, who retain them, to give birth to larval young that then have to spend time in a pouch to continue growing. The presence of epipubic bones in the distant ancestors of placentals has led to suggestions that perhaps live birth began in a marsupial type condition, but evidence for this is hard to come by. In any case, placental mammals now gave birth to much fuller developed or precocial offspring, which could move somewhat more freely, but were still heavily reliant on their mother's care. To date, the evidence suggests that other eutherians and other Mesozoic mammal groups did not have life histories like this, and that this capacity for young to gestate slower but grow faster allowed later placental mammal groups to evolve such massive sizes as elephants and whales within much shorter periods of geologic time. The now extinct relatives of the living placental mammals include a number of curious forms like the leptictids, which walked bipedally like dinosaurs, and the taniodontids, which were specialized digging mammals. But today, living placental mammals are divided into four key groups. The two members of Lantogenata are the Afrotheres, elephants, manatees, hyraxes, tenrecs, and their kin, and the Xenarthrans, anteaters, sloths, and armadillos. Then there are the two members of Boreoeutheria, the Laurasiatheres, which include the ungulates, carnivorans, bats, and a host of small insectivorous forms like shrews, and the euarchontoglirans. Before we jump to them, do you have anything you'd like to add?
No, not really. I think you've covered mammal origins very well here. Yeah, I think it's definitely worth emphasizing that mammals were extremely diverse during the Mesozoic era, as you have indicated here. So there are many lineages that were around then that have not survived into the present day or even past the Cretaceous period. And mammals were extremely successful and kind of made their way into many diverse ecologies. We've actually talked about some of these on the show. You have like, of course, small climbing forms, but also specialized burrowers and swimmers and gliders. Yeah, it's like they are confined to very small body sizes prior to the end Cretaceous mass extinction, but ecologically they're still doing many things. It's a very fascinating period to kind of contrast the different ways of being a mammal before and after the Great Cretaceous mass extinction, which I suppose we will do shortly.
Oh, absolutely. Because indeed, it seems the evidence suggests that most of the living placental mammal diversity evolved only after the KPG extinction, as has been shown as well for crowned birds. But moving on to the next slide, we'll meet one of these groups, euarchontoglirans, which is divided roughly into archontins, which is our ancestral group, and glirs, which includes the lagomorphs, rabbits, hares, and pikas, and the rodents, with over 2,500 species, or a third of all mammals. Archonta, as traditionally defined, includes primates, the gliding colugos, and the tree shrews, as well as bats, but recent work now groups bats with eutheria, despite their anatomical similarities, which are no doubt examples of convergent evolution for tree living. Genetics has typically defined euarchontoglirans, but it has been reported that this clade, among placentals, may have first evolved the appendix for aid in gut immunity, while among the marsupials, the diprotodontids actually evolved independently too. Archonta, meanwhile, evolved a flexible ankle to aid in climbing branches, which is a trait also seen in bats, as aforementioned. In the past, the tree shrews were the odd one out, but genetic studies had flip-flopped on whether they should be allied with the other archontins, allied with glirs, or as an older branch equally related to both. The most recent work seems to recover them as archontins after all, but at the very least, tree shrews resembled what the earliest members of both groups may have looked like. For example, take the stem primates known as Plesiadapiformes, which seem to have evolved by the tail end of the Cretaceous period and lasted for 20 million years. The best known fossil forms of this group, like Plesiadapis proper, show arboreal animals with long tails and a divergent hallux, the bone that makes up the thumb and the big toe, just like other living primates. So we can say that the common ancestor of both groups probably share these features. But it's become apparent that many members of this group do not share features with primates, like forward-facing eyes, which would then invalidate the hypothesis that primates evolved from them. And based on recent studies, like a massive 2022 paper by Anna Wisniewski and colleagues, it seems that most Plesiadapiforms do indeed group together as a stem lineage to the primates, while a few others are better nested as close to the living archontins. In particular, we see the oldest known member of the group, Purgatorius, actually nesting with colugos of all things, while the Mongolian Eocene Atlanteus is a sister clade to primates proper, which we will see on the next slide here. The anatomical characters that define primates consist of, along with a divergent hallux, forward-facing eye sockets for stereoscopic vision, allowing us to see in 3 dimensions, which is good for judging distance among tree branches, a loss of one incisor and premolar tooth in jaws, an elongated calcaneus, or heel bone, for more rigorous grasping of the hands and feet, and claws converted into nails. We also tend to have a sizable brain-to-body ratio compared to other placental mammals, with a large cerebral cortex. The phylogeny of living primates is very well understood, with many questions regarding fossil forms still. We know that there is a major split among primates between the strepsirrhines, or wet-nosed primates like lemurs and lorises, which retain the ancestral rhinarium, and haplorhines, or the dry-nosed primates like monkeys and apes, which gave us an upper lip and so changed the way our facial muscles could move, giving us more expressions to use in our increasingly complex social groups. Haplorhine primates also gained a postorbital plate behind the eye socket, so now the orbit was completely surrounded by bone, which gave protection to these primates' sensitive, now full-color vision. 2 extinct genera of importance to understanding early primates are the stem haplorhines Archicebus and Telhardenia, the latter of which is the oldest known fossil primate. Archicebus was only described in 2013 and is pictured here to the right, would have resembled the living tarsiers, who we will meet on the next slide. So living haplorhines are divided into the tarsiers and their relatives, and the anthropoids, or the monkeys. Tarsiers seem more like lorises and bush babies than close relatives of monkeys, but they share far more anatomical and genetic similarities with us than them.
Hmm.
In fact, despite being mainly nocturnal animals, tarsiers share with other haplorhines the loss of the tapetum lucidum, or eye shine, that many other nocturnal animals have. But otherwise, they are highly specialized primates with their own unique features. The transition to more monkey-like primates can be seen in the successive extinct clades shown here. One particularly important fossil is Eosimius, depicted here to the right, which lived between 45 and 35 million years ago in present-day China. Despite being one of the smallest primates that ever lived, it measured on average a little over 100 grams, Eosimius shows a dental formula more similar to living monkeys than tarsiers. And closer still are the amphipithecids and parapithocoids, which are known from more complete remains and could reasonably be called almost monkeys. The amphipithecids seem to be more specialized for slow quadrupedal walking, but not leaping, among branches and eating hard fruits, while at least some of the parapithocoids were short-faced quadrupedal leapers that ate soft fruits and had a degree of sexual dimorphism. Let's move to the next slide now. Yeah, so for convenience as well as recognition of the phylogeny of living primates, Anthropoidea can be considered the monkey clade.
Hmm.
Feel free to visit episode 2 of the first season to see our discussion of why we chose this. Monkey skulls have eye orbits reduced in size, and so accommodate a far more forward-facing skull than other primates, which may be scaled allometrically to account for a more diurnal or day-living lifestyle in the common ancestor. The tympanic bone associated with the eardrum has also become fused to the wall of the middle ear, and there are changes in blood flow between both the visual and auditory systems. We also find changes in the foot anatomy of monkeys too, consistent with the understanding that ancestral monkeys were still heavily arboreal animals whose distribution and dispersal were heavily tied to tropical forests. Living monkeys are divided into New World or American varieties like capuchins, marmosets, and howlers, and Old World or Afro-Eurasian varieties like baboons, macaques, and colobus monkeys. And as before, scattered between these are a host of now extinct forms like Sedanius, pictured to the right, and the propleopithecids like Egyptopithecus. Now, the Afro-Eurasian monkeys, or catarrhines, have nostrils that face downwards and are compressed down the middle. In fact, the name catarrhine means down-turned nose, and our modern dental formula of two incisors, one canine, and three molars on one side became established as well. Although, if you have your wisdom teeth removed, then that becomes 2122. um, let's move to the next slide now, and here we are. Lastly, we come to the apes, which can be considered a derived clade of catarrhine monkeys. Simply, apes have lost their tails, and based on a recent study from back in February, we suspect that it was simply the insertion of a DNA sequence called an ALU element into the genome of the earliest apes that may have contributed to this loss. The tail vertebrae have been reduced in number and fused into the coccyx, which is still used as an abdominal muscle attachment site. With the tail no longer there for balance, apes use a different type of locomotion in the trees. Now, ancestrally, apes, like the proconsalids, Akembo and Proconsul proper illustrated here, walked quadrupedally on their palms through the trees. As such, their rib cage was very similar to ancestral monkeys. And it was not until later apes evolved that would develop a wider rib cage with the shoulder blades oriented on the back, parallel to the spine, and having a flexible elbow. And this changed how the upper limbs could be moved, giving apes the ability to rotate their arms. And instead of quadrupedal walking, apes were now brachiating, that is, swinging through the trees. We see this very clearly in the oldest living clade of apes, the gibbons and the siamang. Apes underwent a particularly strong period of radiation during the Miocene epoch between 17 and 10 million years ago, and many different lineages evolved and became specialized. Oreopithecines, for example, which lived in the Mediterranean, had such a mishmash of features that they've proven to be hard to classify, and they seem to be slow-moving apes that were not particularly arboreal to begin with. In fact, the classification of all the fossil apes has been rife with difficulties.
Hmm.
The phylogeny shown here is based on a recent attempt with the best evidence available. It seems that the apes traditionally classified as Dryopithecines were stem groups on the lineage leading to the great apes or hominids. And as the name suggests, great apes had larger body sizes than earlier forms, as well as having a more pronounced sexual dimorphism for the most part. Orangutans branched off from the ancestors of the African apes, and gorillas were the next to branch off from the common ancestor of chimps and humans. And all of these non-human ape clades have their own stories to tell, with genetics showing evidence of several ghost lineages and hybridization events that we have yet to find physical fossil evidence for. These types of data make it difficult to pinpoint specific speciation dates for each successive clade, and it's actually now believed that the evolution and spread of the great apes was not so neat as is implied here. Um, but with that, we'll have to pause our journey to the human family tree, where we will pick up in the next episode. But before we move forward with our finale, Albert, do you have any closing remarks about this grand sweep through the tree of life to our common ancestor with chimps?
Well, congratulations for getting to this point. I know how easy it would be to just go on multiple tangents, at every point in this journey. But no, you handled it really well. I'm very impressed. Um, and yes, this is a really great overview of how humans fit into the tree of life. As primate evolution goes, I guess I'll just plug Mike Kesey's comic Paleocene, because that is about primate origins. That's specifically about our primate ancestors right after the end Cretaceous mass extinction. Now, the model that he uses there, like he follows studies that place primate origins a little bit further in the past than is perhaps well evidenced today. So like his early primates, like post Cretaceous primates, are already I think they're already meant to be essentially haplorhines or pretty close. But I think it is pretty likely that crown group primates were mostly a post Cretaceous kind of thing, at least in my opinion and my interpretation of those studies. But no, it's a really interesting piece of work that I definitely encourage folks to check out if you're interested in primate evolution and deep time. So it's a fictional story but told from the perspective of like our primate ancestors. And drawn as a comic series. And something this is not that important, but it does occur to me that like in popular culture, we don't often think of brachiation as an ape thing, right? Like even think of monkey bars. That's what we tend to brachiate on, um, and but we call them monkey bars and not ape bars. And to be fair, as you have explained, apes can be considered technically a kind of monkey, so it's not inaccurate. And also, monkey bars sound better, anyways. But people often apply that kind of behavior to all kinds of monkeys, and no, it is actually specific to apes. So that's just something to keep in mind. It's kind of an interesting observation. But no, I think this was a really great summary. I'm glad you were able to put this together in such a comprehensive but still very well organized manner, and I'm definitely excited to see what you have next in this series.
Absolutely. I'm glad to hear that. Well, let's not keep us waiting longer. Let's move to the next slide. And we'll save our exploration of the hominins proper for the next episode. As we did with the rest of life on Earth, we're going to trace the physical evolution of our lineage, from the last common ancestor with chimps to the point when Homo sapiens evolved. The story in between features a host of players, each a unique and specialized member of its ecological community, with behaviors we can speculate with information, but only so far. We're going to meet Sahelanthropus, Ardipithecus, Australopithecus, and more, and see what the latest research has to say about each. In fact, this will be a much needed update and synthesis to the last season's coverage of hominins, which was only over 3 years ago now. It's almost going to be 4 years. Um, and indeed, paleoanthropology is an ever changing field, so it's fitting that Humanity of Prologue Season 2 is going to cover this topic. And I'm going to do my very best, as I've done here, to synthesize the enormous amount of new data that has come about to see kind of what we know, what we don't know, and what we hope to know, and what we can't know about the evolution of the hominins leading to our own species. But with that, I'm afraid that is the end of our episode. I want to thank you all so much for joining us through this amazing journey. Let's move on now to the next slide to cover our general acknowledgments. We are on Patreon. That is patreon.com patreon.com/time and clades. Patreon is a website where you can send monetary donations of any amount that go to support content creation, and in exchange, you get exclusive updates, news, and previews of content creators' works. So, for example, if you subscribe and donate to our Patreon, we have a tier system where you can see when we have new episodes planned and set record dates. You can see each of the scientific studies we're going to cover or the topics we're going to talk about. You can send in questions. And you can even get shout outs. And so, in fact, we have a tier where quite a number of our followers are owed shout outs. So, we want to thank my sister Julie and our good friends Paul, Denver, Tristan, Frankish, and Baldamunzio for their support of this series. It really means a lot to us. And as we like to say, your contributions will help us continue this series and develop new projects and expansions. So moving on now to the next slide, we want to thank our good friends Henry and Alicia for their contributions to this series. Henry was responsible for the music that opens up each and every episode of Through Time and Clades. And also, I want to give them, very proper thanks for their consultancy, especially regarding the synapsid part of this journey, because Henry works with synapsids. And of course, Alicia is responsible for the color scheme of Albert's Albersaur avatar, a cheery little fellow. We are on Twitter @Time and Clades, where we usually post new episodes as they premiere, but most likely you're watching us on our YouTube Through Time and Clades. So if you wish to like and subscribe, that's always appreciated. If you have any questions about the show, whether about any parts of the journey we've talked about here or anything in general, there are 3 ways to contact us. You can send us an email, timeandclades@gmail.com. You can tweet at us, or you can leave a YouTube comment, and we will respond in kind. And I have a very massive document of references as per each episode of Humanity of Prologue Season 2 that you may access to see many of the papers that were used to support the research that was discussed in this episode. And I've done my best to organize it at least fairly decently so you can follow some of the major transitions in the tree of life and more easily find respective papers for things we've talked about today. And of course, that will be available to view as a Google Doc. But with that, that's it for our show. Again, we have upcoming events and news that we would like to talk about, but I think it's still a little bit too early to say for sure. But thank you all so much for joining us. We wish you a very wonderful rest of your day, and we look forward to seeing you again next time around.
Take care, everybody.